Hello everyone, welcome to the new session of Classification and Nomenclature of Carbohydrates Part 2. So this is the second part and we have to study in this particular session about the pentose sugar, right? We will study about the pentose sugar, structure of glucose and fructose, structure of oligosaccharides and also the structure of polysaccharides. So all these things we are going to cover up in this particular session. So let us see. Pentose sugar. A pentose is a monosaccharide with 5 carbon atom, right? And pentose are organized in two groups. That is the aldopentoses and have an aldehyde as a functional group at position number 1. Whereas the second is the ketopentoses and they have a ketone functional group in position 2 or 3. Right, so these are the two different forms of the pentoses. Either they are aldopentoses or they are having the ketopentoses. Now let us see in detail about the aldopentoses. The aldopentoses have 3 chiral centers and therefore 8 different stereoisomers are possible. So if we are having one single chiral atom, then we will have two different isomers, stereoisomers actually. But if you are having the two, then you will have more the number. But here they are having 3 different chiral atom. So 8 different stereoisomers are possible in the case of aldopentoses. Ketopentoses, 2 ketopentoses have the 2 chiral center and therefore they are having the 4 different isomers. So if you are having one chiral atom, then 2 stereoisomers will be possible, right? And if you are having the 2 chiral atom, then you will have the 4 different stereoisomers. And if you are having 3, then you will have 8 stereoisomers. So in the case of aldopentoses, this 3 chiral atoms are there, so 8 stereoisomers are possible. But here in the case of ketopentoses, 2 ketopentoses have the 2 chiral center and therefore 4 different stereoisomers are possible. The 3 ketopentoses are they are rare, that is the nectarine, saccharine and cyclamate. So this we have already discussed in the first lecture, right? So with this, let us study about the ketopentoses in detail. The carbohydrate may also be classified as either reducing or non-reducing sugar, but it depends upon how they are reducing the Fehling solutions and the Tollin reagents. So these are the two reagents are there. If they are able to reduce these two reagents, they are called as reducing sugar. While if they are not able to reduce the Fehling solution and Tollin reagent, then they are considered as a non-reducing sugar. You must have seen in the table that they are called as a reducing or non-reducing sugar, right? So on the basis of this the reduction of this, or you can say the reducing of Fehling or Tollin reagent, they are considered as either reducing sugar or non-reducing sugar. So all monosaccharides and disaccharides other than sucrose are reducing sugar. So sucrose is one of the example that is non-reducing, while all the monosaccharide, that is the disaccharides and other than disaccharide, they are reducing sugar. So carbohydrates are also said to be saccharides. We have discussed this. So here the structure of fructose and glucose you can see, right? So this is the pyranose ring structure, and this is the furanose ring, right? So here you can see this is the 1 carbon, 2 carbon, 3 carbon, 4 carbon, 4 carbon and 1 oxygen, right? Furanose ring structure. Now here you can see 1 oxygen, 1, 2, 3, 4, 5 carbon and 1 oxygen, right? Pyranose ring structure. And these ring structure can also be found in the form of open straight chain. So this is the open straight chain of the glucose, and this is the open straight chain of the fructose. Now, glucose, that is also called as the dextrose, most important and the common carbohydrate seen in the living world. So this is one of the very important, and you can also say that this is the common type of carbohydrate that we can see in the living world. Means all the animals and the plants, they are found, and it is present. It is the simple sugar of the blood. Like in the blood, the carbohydrate is present in which form? It is present in the form of glucose only. So glucose is transported with the blood, right? And this is the ultimate source of energy in the cellular respiration. So immediate source of energy, this is the thing, that they provide lot of energy when they get oxidized in the cell. So glucose is stored as glycogen. When you are having extra glucose in your body, then that glucose has been transported to which organ? Liver and muscles, and there it is stored as a glycogen. So this is the glucose, and in the ring structure, and this is in the straight chain form. Fructose, this is also called as levulose, most common sugar in fruits. So in the living body, like in the man or the human, they are having glucose, but if you talk about the fruits, then the most common sugar is the fructose, and it also occur in the nectar and honey. It is the sweetest among all the natural sugar, and it is a reducing sugar. So fructose, these are some of the characteristic. Most common sugar in the fruits, found in the nectar and honey, and this is the sweetest among all the natural sugar, and it is a reducing sugar. Derived monosaccharides. Now what are those? Monosaccharides are modified variously to form the number of different substances. Means we can see modification in the case of monosaccharides. Deoxy sugar. So deoxygenation of ribose produces deoxyribose. Means oxygen is removed there and it forms a ribose structure like this, and it is called as deoxyribose. It is found in DNA. Deoxyribose nucleic acid, as the name got. Amino sugar. Monosaccharides have amino group. For example, glucosamine. Right? So amine group, when joined with the glucose, it forms a glucosamine. Sugar acid. Ascorbic acid is a sugar acid. Sugar alcohol, glycerol is involved in the lipid synthesis, is also one of the derived monosaccharides. So these are the monosaccharide which are having some modification in their structure, right? And they are found in the various with the different names. For example, deoxyribose, amino sugar, sugar acid, sugar alcohol, etc. Now comes the oligosaccharides. So bonding of one sugar to another by a covalent glycosidic bond to form a large molecules result in the oligosaccharides. So with two monosaccharide, between two monosaccharide, we know that for the formation of the disaccharide, trisaccharide, tetrasaccharide, right, all type of these saccharide, they are having the glycosidic bond and they form the oligosaccharides. So oligosaccharide contain 2 or 9 units of the monosaccharides, and this type of bond COC linkage is there, and that is the glycosidic bond. So between C1 from one sugar and hydroxyl group from the another sugar, right? So carbon 1 of one particular sugar, right, and with the hydroxyl group from the another sugar, they will have a bond formation, and this bond is known as glycosidic bond. So this glycosidic bond we can see in the case of oligosaccharides. The term oligo means few in number. So oligosaccharides typically consist of 3 to 10 monosaccharide units, and depending on the number of monosaccharides, oligosaccharide can be a disaccharide. Means two monosaccharides are there, and for example sucrose and lactose. Trisaccharides, for example raffinose. Three monosaccharides unite together to form the raffinose structure. Tetrasaccharide. Four monosaccharides unite together to form the tetrasaccharide. An example is the stachyose. We have already seen in one of the figure. The common disaccharides are sucrose, maltose, lactose, and cellobiose. These are the different, you can say, the examples of the disaccharides, right? So, commonly found in the plant and sugarcane, beet roots, carrots, and fruits. So, this is about the sucrose, one of the disaccharide. So, this is found in the plants generally, sugarcane and beet roots. They are sweet in taste because of the presence of sucrose. They are also found in the carrots and some fruits also. The fruits that are sweet in taste. It is the storage product of photosynthesis. So when the photosynthesis when the photosynthesis take place, it takes place by formation of glucose, and it is the storage. It is in the form of sucrose. So plant transport carbohydrate from leaves to other organ in the form of sucrose. This is very important question. Glucose is not transported from the photosynthetic organ, means the organ that take part in the photosynthesis, that is the leaves, to the storage organ. It is transported in the form of sucrose only. So sucrose has no free aldehyde or ketone group, so it is not a reducing sugar. So here you can see the structure of sucrose. They are having the glycosidic bond. So this is the OH group, right? And this is the CH2, right? So one of the H here and OH is utilized to form this glycosidic bond. Maltose or the malt sugar is composed of the two glucose molecules, right? Here both the molecules are glucose. This is also glucose, this is also glucose, whereas in the case of sucrose, one was glucose and another was fructose. So it does not occur in the nature and found during the germination of the starchy seeds. It is not found in the nature, and it is found during the germination. When the germination takes place in the seeds, this maltose is formed, and it is produced commercially from the starch by the starch hydrolyzing enzyme diastase. So we required maltose, right? And maltose can be prepared by starch. So if you are having the starch, with the help of diastase enzyme, it is converted to the maltose, and maltose is used in the liquor industry for making beer, right? So this is maltose. This is very important. Next is the lactose, another disaccharides formed by the condensation of glucose and galactose. So glucose and galactose, one monomer is the glucose and another is the galactose, and lactose hydrolyzed by the enzyme lactase, and this is the present in the plasma membrane of the cells that line the intestine. Means this lactase enzyme is present in the plasma membrane of the cells that lies in the intestine. So they hydrolyze this lactose by the enzyme lactase. And many people lose this enzyme at the childhood and find that eating dairy product cause digestive discomfort, because this lactase enzyme is not present in their cells, right? That is present in the intestine. So they are not able to digest that lactose. and that is why they are called as lactose intolerant. Functions of small carbohydrates. So triose, glyceraldehyde, dihydroxyacetone are important intermediates of both respiratory and photosynthetic pathways. So if you study about the photosynthesis or the respiration, you will see triose phosphate, glucose triose phosphate or fructose triose phosphate is formed. Glyceraldehyde is formed. Dihydroxyacetone phosphate is formed in the glycolysis. So all these are the intermediate that are found in the respiratory and photosynthetic pathway. So how this monosaccharide, disaccharides are important, you can understand, because they are the intermediate of the two very important physiological pathway in the plants, that is the respiratory pathway and the photosynthetic pathway. Pentose sugar, arabinose, xylose form the polymers that are constituent of the hemicellulose. Now, again, the hemicellulose is a structural component of the plant, right? And they are formed by the pentose sugar, arabinose, xylose, and they form the polymers. And they are the constituents of the hemicellulose. Ribulose 1,5-bis phosphate, very important, the acceptor of carbon dioxide in the photosynthesis, is also ribulose 5-carbon compound monosaccharides. In some plants, oligosaccharides serve as a reserve food, that is sucrose. Sucrose, it is a form of a reserve food. Fats and amino acids are formed from the glucose and other sugar, right? So fat and amino acid, when lots of glucose is present in the body, then it is converted to fats and sometimes amino acids also. Oligosaccharide attached to the cell membrane from cell coat or the glycocalyx. So in the case of bacteria, the outermost covering is the glycocalyx, and it is made up of oligosaccharides. Ribose is an important pentose sugar which is found in a variety of chemicals like coenzyme A, FAD, NAD, NADP, and also the ATP. So ribose is a very, very important, you can say, the carbohydrate that is present in the coenzyme A. So if you remember the acetyl coenzyme A, the first compound that start the citric acid cycle, that get enter in the citric acid cycle, acetyl coenzyme A. FAD get converted to FADH2 in citric acid cycle. NAD and NADP reduced coenzyme, and ATP the currency of energy, ATP. So all these important chemicals that are present in the cell, right, and they are very important, found in the most of the physiological pathways. So it is made up of ribose sugar. Polysaccharides, they are formed by the polymerization of large numbers of monosaccharides, that is monomers. They are also called as glycans. They are long chain, right end is reducing end, whereas the left end is the non-reducing end. The chain may be branched or unbranched. So sometimes they are branched and sometimes they are unbranched. They are three main types: storage, structural, and mucopolysaccharides. So three types of polysaccharide we are having, right? So food storage polysaccharide, they serves as a reserve food material. And examples are starch, glycogen, and inulin. These are the two main storage polysaccharide, that is the starch, glycogen, and inulin. Starch, we will study about the starch. It is the storage form of the polysaccharide in plants, and it is a polyglycan homopolysaccharide. So one type of monomer is there. So they are stored in the chloroplast or the special leucoplast called as the amyloplast. So there they are get formed, and they occur in the form of microscopic granule that is called as starch grain, starch right? They are found in the form of starch grain in the microscopic. You can see in the microscopic microscope, and they are found as a microscopic granules, and then it is called as starch grains. And starch consists of two components, that is the amylose and amylopectin. Now, what are amylose? Amylose is a more soluble in water than amylopectin, and it gives dark blue black color with the iodine. Starch form the helical secondary structure, so it can hold I2 molecule, and that is why it gives, when it hold I2 molecule, it gives blue black color, right? So this is the also used or famously called as the starch test. So here you can see the structure of the amylose, and this is the structure of the amylopectin. So amylopectin is forming the branch chain, whereas the amylose, it is having the straight chain. And here you can see alpha 1,4 glycosidic linkage is here, and here it is alpha 1,6 glycosidic linkage at the branching point. Amylose is in the form of the continuous straight but helically arranged chain, where each turn contains about 6 glucose unit, and the successive glucose unit are linked by 1,4 alpha linkages. Whereas the amylopectin contain large number of glucose unit, and this is 1,3 linkage in the case of amylopectin. Now comes the glycogen. So here you can see the glycogen. It is a polysaccharide food reserve of animals, bacteria and fungi, and it is popularly known as animal starch. And it has about 30,000 glucose residue, that is a molecular weight around 4.8 million. So these are the monomers, right? And 30,000 glucose unit unite together to form the glycogen molecules. Glucose unit are highly branched and brush-like chains, and it is 1,4 alpha linkages in the straight part, whereas 1,6 linkages in the branching point. And the straight part is helically twisted, which each turn having the 6 glucose unit. So here you can see the structure of the glucose. This is the amylose and this is the amylopectin form. Next is the inulin. Inulin is a polymer of fructose, and it is a storage polysaccharides of roots and tubers of the dahlia. So dahlia plant, they are having the this inulin as a storage polysaccharide, and it is not metabolized in the human body and filtered out from the kidney by the excretion. So it is used in the testing of kidney also. Next, structural polysaccharides we can see in the skeleton and some plants like fungi, right? In fungi, and it is also found as a skeleton in some animals. Chitin, that name is chitin. So this is the second most abundant organic substances after the cellulose, and the structural polysaccharide found in the exoskeletons of some arthropods, that is the insects and crustaceans. Their exoskeleton is made up of this chitin. And also the cell walls of fungi, they are also having this chitin. Last is the cellulose. It is the most abundant organic substances present in the biosphere, and the cellulose is a linear polysaccharide, but it is consist of repeating unit of beta glucose linked together by the beta 1,4 glycosidic bond, and it contain around 6,000 glucose unit residue. So here these are the fibers, and this is the one of the cellulose structure fiber you can see, and this is the you can say the monomer, that is repeated glucose unit. So they are the more complex polysaccharide in nature, and they have the building blocks amino sugar and chemically modified sugar, that is the glucosamine and N-acetyl glucosamine. This is found in the bacteria cell wall, and they are the modified sugar. Glucosamine is the natural compound found in the cartilage, and N-acetyl glucosamine is the necessary for the intercellular communication, and it is concentrated in the sensory nerve structures of both human and in animals. Last type of is mucosubstances. So they are slimy substances or the mucilage which possesses acidic or the animal polysaccharides, and they play an important role in lubrication, protection, moistening, facilitating the movement and trapping substances. And some examples of the mucosubstances are agar, pectin, hemicellulose, peptidoglycan, and the mucoproteins. So this is all about this session. Thank you very much.